we actually cover a whole gamut of trying to, I would say, break one of a key code that pancreatic cancer cells uses to survive. In the incubator are a number of, and don't be scared here, are a number of tumor cell lines that are growing from patients that have long passed on. But yet their cells, the tumor cells, are still growing as long as we put them in a condition that fits somewhat physiologically the way the human is. Why are these cells able to survive so much? Why are they so resistant to the therapies we give them? And we believe that there is a specific stress response protein or pathway, we call it HUR, that's very important in different ways of keeping these pancreatic cancer cells superhuman, if you will, and make them immortalize them, allow them to succeed and thrive in very stressful conditions. Because even when a patient has a pancreatic tumor, sometimes that microenvironment can get very deprived of oxygen, deprived of glucose, deprived of things to grow, but these cells seem to survive. And then we treat them with these very toxic agents, and they're able to resist and survive. And basically, the lab, in brief, is really trying to figure out ways, one, to understand how they're able to survive through this pathway, the stress response pathway, and two, had to actually go in there and disrupt that so we can literally hit the pancreatic cancer cells at their knees so they can't survive and thrive. And this pathway is almost like a seatbelt for them so they can get through the tough times and survive, and we want to be able to cut that seatbelt and really get rid of their airbags. When money comes directly to this laboratory, each dollar makes a huge difference. Each dollar goes directly to what you see around here. All these reagents cost significant money. And for almost every reagent and every experiment, we learn something. Even $50, $100 can make a big difference towards one experiment being done. And that difference between $50 and $100, or even $1, that adds up, can make the difference of whether we be able to run an experiment or not run an experiment.